Good morning. I'll be talking about microbiome of oral biofilm. So first we'll talk about what is biofilm, its formation and architecture. Biofilm is a layer of microbes that multiplies and colonizes on or with the body part. Biofilm properties: it is spatial and functionally organized, enclosed within a matrix of extracellular polymers and tolerant to antimicrobial agents and increased pathogenicity. Biofilm visualization, 3D images with confocal laser scanning microscopy helps to visualize or microbe locations specificity with fluorescent in situ hybridization can be used. Biofilm growth, environmental limiting factors and such as nutrient pH, oxygen changes can affect the growth of biofilm. The effect of biofilm presence. If the biofilm is present, it can have direct or indirect effect. The direct effect is expression of subset of genes due to the surface attachment of biofilm with the tissue, or indirect effect can be expression of different genes due to the changes in the environmental of the environment around the biofilm. So, how does biofilm get antibacterial resistance? The phenotypic resistance is due to the dense matrix limiting in-depth drug interactions, horizontal gene transfer within the colonies promoting bacterial resistance via plasmids, less drug susceptibility due to the slow rate of microbial growth in matured biofilms. Production of drug neutralizing and drug degrading enzymes by the bacteria of the biofilm also affects the causes antibacterial resistance. So in the oral cavity, the biofilm is called as the dental plaque. It is microbial growth and colonization forming a layer on teeth and gingiva. It is characteristics features of a typical. It has the characteristic feature of a typical biofilm. Calculus is the calcified form of plaque that happens over a long period of time. Plaque presence, it is demonstrated by disclosing agent, which can help to indicate the color change when in the presence of plaque. Then location, it is naturally present on stagnant surfaces of oral cavity, for example fissures, proximal surfaces, gingival surface, denture, tongue, and desquamation prevents the bacterial adhesion on mucosal surfaces. The beneficially function, it provides resistance to exogenic colonization of pathogenic microbes. So coming to the mechanism of plaque formation, it is a dynamic and continuous process of attachment, growth, removal and reattachment of bacteria. So first it is acquired pellicle formation. The pellicle forms on a clean tooth surface by salivary phosphoproteins and glycoproteins, including stadtherin, proline-rich peptides, and host defense components and lipids interaction. The salivary interactions with enamel surface forms pellicle on lingual surface of size 20 to 80 nanometers, and on the buccal surface of size 200 to 700 nanometers after 2 hours of exposure. The bacterial and host enzymes such as amylase, lysozyme, carbonic anhydrase, glutens, gingival cervicular fluid. Fructosyl transferase and glucosyl transferase are involved in the formation of pellicle. Next, we'll see about the microorganism transport and reversible attachment of the microbes. The passive transport of microbes occurs by salivary flow, and the active motility is present in some bacteria. As we can see with the graph, the weak long-range electrostatic active forces are followed by strong short-range bacterial adherent surface receptor molecular interactions. So initially there is the VT line on the graph shows that there is a repulsion followed by attraction as the time passes. So the presence of negatively charged on pellicles and bacterial surfaces cause weak repulsion, and strong ionic strength of saliva causes bacterial adhesion on the pellicle. Talking about the pioneer microbial colonization and permanent attachment. So increase in short-range physiochemical interactions of pellicle molecules and adhesins on the bacterial cell surface makes the attachment permanent. Hydrophobic surface component of cell dehydrates the water film, increasing the short-range interactions, leading to the permanence of microbial attachment. Bacterial appendages show adhesins that increase cell-to-cell or cell and surface interaction. Pioneer polarization, mitis group of streptococci survives by producing IgA1 protease that provides immunity against the host, and actinomyces, SPP, hemophilia, Neisseria with low obligatory and anaerobic species. Streptococcus mutans, Porphyromonas indivis, Promotella, Luishi, and other species are like rich in proline on peptide surface. So Streptococcus sorbinis colonization is more dependent on the sucrose mediated mechanism. Streptococcus sanguinis can bind to terminal sialic acid residues in absorbed salivary glycoproteins. Streptococcus oralis expresses lectin that interacts with trisaccharide structure containing sialic acid and galactose. S. gaudini can interact with both salivary proteins and nocilin, which is the co-aggregation between the two bacteria. And actinomyces, SPP, different types of fimbriae are present for adherence to the PRPs and staterin, and for adherence to attached bacteria and buccal surfaces. Then coming to co-aggregation and co-adhesion and microbial succession. So increased diversity with increased actinomyces and gram-positive bacilli proportion. The attachment by adhesion and the attachment by adhesion of bacteria to pioneer species. The pioneer community is the first colonizing community. So the metabolism of that community alters the local environment for growth and growth of fastidious bacteria. The O2 intake and CO2 release from pioneer community favors the growth of obligatory anaerobic bacteria. The metabolic end products of the pioneer community are used as the primary nutrient source for other bacteria. So co-aggregation is the cell-to-cell recognition of genetically distinct cell types and is important for the biofilm formation and microbial succession. Early plaque formation by Streptococcus and Actinomyces shows co-aggregation with further intrageneric and co-aggregation of primary colonizers and other genera. Development of food chains also happens among the bacteria, which is facilitated by co-aggregation of oral microbes with each other. Lectin mediated interactions between streptococci and actinomyces can be blocked by adding galactose or lactose or treating the receptor with protease. Fusobacteria co-aggregates with the widest range of bacteria with no self-aggregation but acting as an important bridge between early and late colonizers. The early colonizers co-aggregates extensively with Fusobacterium, whereas the late colonizers such as Salmonella, Prevotella, Eubacterium co-aggregates with Fusobacteria and not with the early colonizers. As we can see in this image, the Fusobacterium acts as a bridge between the early and the late colonizers. Then maturation of biofilm and matrix formation. Increased species diversity due to microbial growth and succession is seen. Significant bacterial growth rate reduction with biofilm maturation. Then the biofilm maturation occurs by the development of extracellular matrix of polymers such as soluble and insoluble glucans and fructans, proteins and extracellular DNA via lysis of cell signaling. The enzymatic degradation of eDNA can weaken the biofilm structure, but the DNA producing bacteria can digest the eDNA to form nucleotides. The biofilm matrix is a chemical scaffold that maintains the structural integrity and the general tolerance of environmental factors and prevents the antimicrobial penetration. The vertical and horizontal stratification can create the environmental heterogeneity, creating a mosaic of microenvironments and changing species distribution. The biofilm architecture, it is palisading regions of filaments and foci aligned in parallel and at right angles to the enamel surface with microcolonies of single cell population. It plays a direct role in host biofilm interactions as the outer layer of subgingival biofilms resemble neutrophils. Lastly, the detachment from surface. So shear forces can cause detachment with some having active self-detachability by proteolytic enzyme cleaving the cell surface proteins. So the changes in environmental conditions are sensed by these bacteria, promoting gene expression and involves active detachment of the bacteria. Now the consequences of plaque formation.